My name is Ansen Tan, and I am a senior at the University of Michigan studying biomedical engineering. Today, I will be presenting my engineering honors capstone project titled An Examination of the Ovarian Extracellular Matrix Composition in Reproductive Age Women. So here is my poster in its totality, and we will begin here on the top left, where I can provide some relevant context to understand this project. So mammalian reproduction can be boiled down to the fusion of a male reproductive cell or a sperm with a female reproductive egg or ovum. The precursor to the ovum is called the ovarian follicle, otherwise known as the functional reproductive unit of the female reproductive system. While men will begin sperm production during puberty and will continue to generate sperm for the rest of their lives, women are born with all of their ovarian follicles at birth and do not have the ability to create more within their lifespan. So prior to puberty, ovarian follicles are preserved in a primordial or dormant state. This is also known as quiescence. Afterwards, the female body selects a handful of primordial follicles to activate and undergo a process called folliculogenesis, otherwise known as the maturation of ovarian follicles. Once activated, the primordial follicle will transition into a primary, then secondary, and then finally antral follicle, which will release the ovum once it's fully mature. And once a woman's follicle reserve has been sufficiently depleted, they enter menopause. While the female reproductive system and folliculogenesis has been greatly studied, how exactly the body determines which quiescent follicles to activate is still largely mysterious to the field of women's reproductive health. The ovarian tissue can be rudimentarily separated into two components, the cells, which are the germ and somatic cells, and then the extracellular matrix, or ECM. The ECM is a complex network of proteins and molecules that provide structural support, organize, and connect cells within each other within the tissue. Interestingly, it has been shown that the ovarian extracellular matrix plays a significant role in signaling pathways, mechanical cues, and is responsible for regulating follicle health. This study seeks to investigate the differences in ECM composition between different regions of the human ovary, as well as how the compositions vary with respect to age. So the human ovary can be compartmentalized into two distinct regions. Here you can see right here in figure 1A, and that is the cortex and the medulla. The cortex represents the thin outer layer of the ovary, which contains the ovarian reserve. And once activated, they migrate towards the center of the ovary or medulla, which houses growing follicles. So achieving a greater understanding of this process will directly contribute to the field of women's health. And this is because the female reproductive system plays a profoundly important role in regulating bodily functions and overall well-being. Hormones such as estrogen and progesterone are not only contribute to folliculogenesis, but also play a pretty significant role in maintaining bone and skin health, mood and temperature regulation, and other important bodily functions. Young women who undergo cancer treatments can often suffer from premature ovarian insufficiency, which is one of many conditions that impairs the ovary's ability to function regularly and activate quiescent follicles. The significance of this project will hopefully establish the groundwork for future investigations into the ovarian ECM composition and its role in the activation and selection of quiescent follicles. If the mechanisms behind follicle activation can be completely understood and controlled, then damaged or abnormal ovaries prior to menopause can be treated and normal hormone production can be restored in order to maintain healthy bodily functions. So in this investigation, we're going to be looking at 6 essential proteins in the ovarian ECM, and they are right here, this list right here. It's 3 collagens, so collagen 1, collagen 4, collagen 6, fibronectin, laminin, and prolactin. And these are all essential proteins within the ovarian ECM shown ubiquitously throughout the cortex and medulla. So I'll be gathering my data from 5 donor tissues and staining against these 6 proteins using antibodies in order to measure their fluorescent intensity, which will tell us how concentrated or how present these proteins are within different regions of the ovary. This project can be broken down into 2 main components, the immunofluorescence staining, which you can see on the left, and if we go down, the fluorescence quantification. So ovarian tissue samples were harvested and fixed in PFA. Then they were sectioned using the microtome, and each section contains fragments from the cortex and medulla. And with a total of 4 rounds of staining, each section will be stained using these antibodies alongside DAPI, which targets nucleic DNA fixed in cells, so that we can visualize the total number of cells within the defined region. So after several rounds of staining, we're going to be using confocal microscopy in order to visualize the intensity of fluorescence that each of these proteins exhibit once a certain wavelength of light is exposed to these tissues. So in figure 1A, this is the overview process of our methods. We're starting with the ovary, we're sectioning it, staining it with antibodies, and then imaging it under confocal microscopy. 1B is one example of a microscopy figure. Here is the medulla and then the cortex, and these are stains for DAPI, laminin, and fibronectin, and their colors are respective. Afterwards, each image acquisition is exported to the Leica software, LASX, for image, or exported from LASX software to ImageJ for quantification. So fluorescence was quantified by converting each individual fluorescent channel into its own unique grayscale image in order to afford the mean intensity of fluorescence and the total area of intensity for each replica. Then we will count the number of nuclei across the entire image by converting the DAPI channel only into a binary image with a 200 pixel size discrimination. So once the data is collected, we can plot these values against the donors or the regions of interest or proteins of interest to see what we can discover. And so we're going to move into the middle portion of my poster. So this first figure is pretty big. And so basically what this is that I calculated the average mean intensity using the values that I got from ImageJ and then plotted them against each donor in each region of interest. And this was done using the PRISM software. So each graph represents a single protein of interest alongside a picture on the right that shows the stained image with DAPI. DAPI is going to be in blue. All the proteins are either going to be in red or green. So from these pictures, from these images and figures and graphs, we can see that there actually isn't much that we can take away from just looking at the proteins individually. What we can see is like the collagens, collagen 1, 4, and 6, and fibronectin are similarly present in the ovarian, medulla, and cortex. Additionally, perlecan and laminin were a little more prevalent in the medulla in nearly all cases. This could be because that these proteins are largely involved in angiogenesis, which is the development of vasculature, mechanical stability, and cell proliferation and differentiation. And this is probably because they're found probably more in the medulla, from my hypothesis, since the medulla is highly vascularized and is responsible for the development of activated follicles. Proteins such as fibronectin drastically changed in intensity, while others, such as the collagens, were consistently observed throughout the cortex. So while these graphs may not give us a clear explanation as to what's going on, the next figure shows something a little bit more interesting. So here, instead of comparing the average mean intensity to the individual proteins, I took a more holistic approach and compared the total area of intensity for each ECM component in the medulla and cortex and put them into one graph. And here we can see that the ECM composition is actually relatively similar between the medulla and cortex regions of the ovary. And then when we normalize this data, which is graph B, we see very similar results. And when we normalize it, this is just adjusting the data respective to the number of nuclei. So these graphs seem to indicate that the ECM composition is relatively homogeneous between the cortex and the medulla when comparing it against each other if we're only looking at these 6 essential proteins in the study. So next, we're going to go to the top right here. Yep. Let me move this. So next, I compared the ECM composition of each protein in each region of interest with respect to age and reproductive age women. While the lack of ECM composition changes, or while there is a lack of change in ECM composition, this somewhat supports the implications from the previous figure that we just looked at. However, these differences cannot be confidently stated since only one of each donor from each age was used in this experiment. So this was just something that I thought was interesting, and I thought that would be cool to put in this graph, but it doesn't definitively say much. So while a comprehensive analysis of the ovarian ECM composition across the reproductive lifespan has yet to be published, but this study provides like a brief insight into the differences in ECM composition between the medulla and the cortex, as well as discrepancies with respect to age. So my findings showed that the medulla and cortex, with respect to these 6 proteins, shared relatively similar amounts of ECM. These results showed that these results could indicate several hypotheses. For one, that these are the essential proteins necessary for structural support, vascularization, proliferation, which may suggest that other niche, less ubiquitous components of ECM could play a more crucial role in follicle activation. Also, this study examined ECM composition of healthy reproductive age women due to limited resources. In the future, we could look at the ECM composition across a greater age range, looking at women who are prepubescent, perhaps women undergoing puberty, and then women undergoing menopause and in menopause. Those changes would probably be more drastic. Furthermore, another interesting direction that we could take includes looking at the ECM discrepancies with respect to race. I tried to do that, but in our study, 4 of the 5 donors were Caucasian, meaning that any comparison to the single non-Caucasian donor wouldn't result in sound conclusions. So the application of tissue engineering within the field of women's reproductive health is a really unique approach to explaining how the female body regulates its ovarian reserve. Just recently, in the past few decades, we saw a huge influx of research looking at the intersection between cells and their surrounding ECMs, looking at the tissue as a whole rather than just exploring these components separately. So while this study may not have produced any definitive answers to the questions I was initially asking, it does serve as a proof of concept experiment that future researchers can build upon. So that is my presentation. I would like to thank the Shikhanov Lab and their members for providing me with the resources and all the help throughout my 3 years there. One of the biggest reasons why I wanted to look at this specific component of my research was that I was very inspired by a lot of the PhD students and people working in that lab. I never thought that I would be interested in this kind of stuff, but it's actually very fascinating. And I'm glad that I went through this process of in my own research and asking my own questions and coming to my own conclusions.